Deepak won't be able to see them as he's presenting, but there's many of us who are somewhat knowledgeable who can have a go at answering questions as they come up and maybe kind of field questions during the Q&A session.
Okay, and Nicole is posting and Nicole is recording. It looks, yep, so we are good to go.
Yes, sorry about that.
Thank you, appreciate that. Thank you for reminding us that the recording was not working. So appreciate that. So where was I? Yeah, about ontologies. So there are several ontologies both in life sciences and outside in the industry, if you look at it, and they're all domain-specific ontologies built for specific purposes. And in biology, the most common ontology that everyone might know is gene ontology for gene function. And if you look at the open biomedical ontologies or the OBO Foundry, you would find a whole lot more examples like the Mondo disease ontology, human phenotype ontology, and so on. And the other terminology that I want to address is a knowledge graph. And there are many definitions, and it takes some time to kind of accumulate a more sensible definition of what a knowledge graph is. But the one that I've found to be easiest to explain to others is it is a graph that represents knowledge, where entities are represented as nodes, and relationship between these entities are represented as edges. So in a knowledge graph, the relationship between entities are first world entities or first class citizens, and that allows you to do pretty clever things and represent knowledge in a more intuitive manner. And so knowledge graphs collectively sort of refers to the property graph style of representation, for example, Neo4j databases, and also RDF graphs, which can be queried using Sparkle. And knowledge graphs have been around for a while. The idea of using a network for representing knowledge has been there since the 1970s with the research into artificial intelligence. And more recently, Google in 2012 kind of brought the term knowledge graph as part of their search. And that's when it started getting more traction in the industry and also in the academia, where people really started looking into how graphs can be used as a store of information for a lot of the clever things that they are doing today. And this is sort of a survey done by Gartner Research, which is a research firm that looks at technologies and their trends. And we see that, and the plot is the expectations for different technologies across time. And we can see how knowledge graphs are like at the peak of the hype cycle currently. So you'll keep hearing more and more about knowledge graphs, and people will try, and companies will try to bring them up and try to build more tools on top of that works with knowledge graphs. And you will see that even in life sciences. So there's a possibility that you will hear more about knowledge graphs and a proliferation of tools that allow you to build these knowledge graphs. And even to pick some examples, even there are several biomedical knowledge graphs, or KGs in short. And for example, the semantic medline database is probably the first example of knowledge graph. And then we have HetioNet, Wikidata, the Monarch Initiative, the Scalable Precision Medicine Oriented Knowledge Engine that is spoke at UCSF. And more recently, in this year, we've had more COVID-19 specific knowledge graphs like the COVID-19 graph and KG COVID-19 and so on. So there are many names out there. And why do people do this? Like why use knowledge graphs? Is because it allows you to flexibly represent data and knowledge. When you try to, it's easy to decompose complex knowledge or complex representations into, when you're trying to represent those information as graphs. And knowledge graphs that have ontologies and all the ontologies loaded in them allow for deductive inferences through logical rules or axioms, where you can, based on existing edges in that graph, you can predict new edges. And there are tools that allow you to do that. And more recently, KGs have been useful for graph-based machine learning, where you can use them as inputs to neural networks, and they can be used for edge prediction or mode classification, which are very useful applications in life sciences. And they also serve as a reliable store of information for downstream applications, tools, and APIs. And so, which is the reasons, one of the very few reasons why we see more of the adoption of knowledge graphs. But there are a few challenges. Note that when groups build knowledge graph, they're not built for, they're built for a specific task that they have in mind, and and it's sometimes difficult to use them in a context for which they weren't originally designed for. And this leads to some incompatibilities, and this incompatibility can be due to a difference in the formalisms used to represent nodes and edges. For example, one graph may be in a property graph form where you have properties on edges that describes those edges, where, and for example, Neo4j. But then there can be RDF graphs where you cannot really do that. There are ways around that, but then that would require you to transform the entire graph, which is not which is a sort of heavy lift to do so. And another challenge is that the vocabulary used may be different between graphs. For example, and this difference can be either syntactic or semantic. If it's syntactic, it could be something as simple as one graph calling a gene an uppercase gene, and another graph calling a gene the lowercase gene. And these changes can get in the way of actually querying and sort of integrating and federating across these graphs. And the same is the case with edges. Then KGs typically lack schema, and that's sort of the main reason for using graphs is because you don't need a schema, unlike SQL databases. But at the same time, that can be a problem because now you don't know ahead of time how that graph is structured. And there are groups that would provide that schema ahead of time, and sometimes in a machine readable form, but not all the time. So it becomes difficult to know ahead of time that what is it, what is the structure, what is the vocabulary that graph is using. And the choice of identifiers across graphs can be different. For example, just to take the human gene, there can be at least 4 different ways of referring to the same human gene. For example, you can use the NCBI gene identifier, the ensemble identifier, a GNC identifier, or even gene symbols. And so these differences become, these different styles of representation makes it difficult to work with the same entity across graphs. And the modeling decisions used to represent data from various sources can be different across graphs. For example, one group may transform a particular data source in one particular way, and another group may do it in a slightly different way that sort of breaks how you try to integrate these 2 graphs together. So these are some of the challenges that we did see as part of a project called the NCATS Biomedical Data Translator. And this is a project funded by the National Center for Advancing Translational Sciences. And the goal is to build a system that is capable of taking existing biological and biomedical data sets and translating them into insights. So it's sort of building a system that is capable of understanding a query from the user, and then the system will then query existing knowledge stores and then give back the results, ranking and scoring them as appropriate. And it's a multi-site, multi-year project, and there are 14 teams spread across the US and another team in Netherlands. And each team has one of 3 roles. Either they are a knowledge provider. These are groups that actually do the heavy lifting of building these knowledge graphs. We have autonomous relay agents that are tasked with taking a query and then querying these knowledge graphs. And the autonomous relay service is what is sort of a user-facing service that takes a query from the user, translate that into a query that then the relay agents can understand. But this is sort of an overview of the architecture, and this has changed a bit across time, but it still, in principle, it remains the same, where the flow of information is going from right to left, where we have different knowledge sources, and each knowledge source is sort of specific in the type of information that they provide, or the data types that they provide. For example, certain sources may be rich in gene information about genes, some may be rich about variants, functions, pathways, disease models, and environmental exposures, and so on. So the idea is that we would create an integrated knowledge graph such that these graphs can be queried by these relay agents, and then the results can be ranked, scored, and prioritized, and then sent back to the user. And the user can be a student, a researcher, a technician, a clinician, or even a scientist, what have you. But to achieve this goal, we as a consortium had to agree on 3 things. One is a shared data model for representing knowledge across the consortium. The second is a shared specification for exchanging information between these tools and APIs. And the third is a shared set of tools that can be used across the consortium so that we are not reinventing the wheel. And this shared data model is what we know as the Biolink model. And while the model was built to serve the needs of the translator consortium, it still has, we are still trying to address the needs outside the consortium in other projects, which I will get to in subsequent slides. So the model itself is a data model for representing biological and biomedical knowledge. It bridges across multiple vocabularies and ontologies. And it is agnostic to the sort of graph representation that you use. So you can use either property graphs or RDF graphs. As long as you're using the vocabulary from the model, there are enough toolings built to work across those differences. So the model is structured into 4 parts. We have entities, associations, predicates, and properties. So entities are represented as nodes in a graph. They represent entities found in biological knowledge and also literature. And they are arranged in a hierarchy. And the root of all entities is what we call the named thing, which can be any entity that exists in a database. And examples of these entities are either gene, protein, disease, phenotypic feature. And each entity has its own unique stable URI, mapping to other ontologies, and a list of valid ID prefixes that can be used to represent nodes with of that entity type. So here, in the case of gene, which is a very common example, we have a URI Biolink gene, and mapping to ontologies like sequence ontology, the semantic science integrated ontology, and Wikidata. And also a list of valid ID prefixes that can be used for identifiers of this type, with NCBI being the highest priority, and then the list goes down with decreasing priority. And that's an NCBI gene being the top because it's the most common sort of namespace for identifiers across organisms. And these entities or these entity classes are basically higher level terms that can be used to categorize nodes in a graph. But if you want more detailed typing, you can always use terms from specific ontologies that further specializes what that type of node is. Now, the next part is the associations, and they represent edges in a graph. Associations are basically assertions or statements that are made by knowledge sources, where an assertion kind of it connects 2 nodes in a graph together based on a predicate or a relationship. And an association gives in the model itself, we have a hierarchy of associations, with the root of all associations being the association class. So for example, a gene-to-gene association would link 2 genes together, and the relationship of that association will tell you the nature, what is that link between the 2 gene nodes. So as I said, the technical term is that an association connects a subject node or the source node with an object node, the target node, together via a predicate. And the nature of the association is defined based on its properties, and association can have additional properties like provided by, evidence, and publication that talks more about where that association came from, what are the evidence that's supporting that association. And certain associations can have additional properties. For example, the disease to phenotypic feature association has a property called the frequency qualifier that represents the frequency with which that phenotype is observed in a population with that particular disease. And the next part is the predicate hierarchy in the model, which it consists of relationships or predicates or relations. There consists of a hierarchy of relationships that can be used as a predicate in an association or a statement. And these relations or predicates are usually mapped to other ontologies, most commonly relations ontology, because most of our use cases are sort of addressed by relations ontology. So bringing this all together, I think, so what Biolink Model gives us is a common dialect for representing knowledge across the project. It bridges across Neo4j graphs and RDF graphs. These are sort of 2 common representations that we have in the project. And we map to existing models, and we ensure that we are curating this mapping as the model evolves. And what the model allows us to do is have a shared space for discussion on more complex representations, like how you would go about representing evidence, provenance, context, and confidence for a particular statement or assertion. So in terms of the curation process, weekly data modeling calls where we bring in, where members of the consortium come up with their concerns or their needs, and we make sure that we're work towards refining the model. And we also make sure we follow a set of well-defined guidelines for governance, where we make sure that new changes are linked with the release, and we communicate these changes to the users and stakeholders. And we also set up weekly help desks for users, where users can bring their data, and we make sure we work with them on how that data can be modeled. And sometimes it can be a two-way street, where maybe the model is not flexible enough, then at which point we extend the model to further accommodate their that particular data type. But more broadly, we want to support the use cases of the wider community outside of the translator consortium, and which is what we hope to achieve today as part of this workshop. But it's not enough to just have a model in isolation. We also need tools that can be used to work with the model. And the first tool is sort of it's a bit more meta, where the first tool is the Biolink ML, which is the modeling language. And then, this is separate from the Biolink model that I mentioned in the previous slide, because the Biolink modeling language is a modeling framework which is used to build the Biolink model, where we have YAML as a source of truth. And everything that is particular to a model is defined in that YAML. And then from this modeling framework, we generate things like JSON schema, Python data classes or Java classes, GraphQL API, GraphQL definitions, and JSON LD context for RDF to JSON serialization, and RDF turtle, and even shape expressions for validating RDF graphs. But just to give an overview of the modeling language or the framework, everything in the model is sort of defined as either a class definition or a slot definition. And we also have schema definitions, and we can import other schemas into the model using Biolink ML, and everything is basically an element. And the modeling framework supports polymorphism, so you can have mix-ins, and you can also inherit properties from other classes. And we borrow a lot of semantics from OWL, because while OWL's style of modeling is a bit more formal, we wanted something of a middle ground where we can define a schema, but yet bring the good parts from the OWL in terms of, say, having domain and range constraints on certain properties, which allows us to say what values a particular field can have, and so on. The next tool is the Biolink Model toolkit, which is a Python API for working with the model. It provides convenience methods for querying the model. For example, if you want to find a Biolink class that, if you want to come in with an external ontology term and then find a Biolink class that is mapped to that term, the Biolink Model toolkit is what you would be using to do that lookup. And the third tool is the knowledge graph exchange. So I'm trying to bring all this together, where we spoke about knowledge graphs and spoke about how Biolink Model can be used as a schema for representing knowledge graphs. But what KGX does is a Python library that allows you to work with knowledge graphs, where you can exchange graphs and also you can convert graphs from one form to another. KGX is compatible with Sparkle RDF graphs or Neo4j graphs, CSV, TSV forms of knowledge graphs, JSON, some parts of all, and even ontologies that are in OboGraphs JSON format. And the features of KGX is that it allows you to transform graphs from one form to another. So you can go from Neo4j graphs to RDF graphs or the other way around. And you can create graphs or subgraphs that are specific to your use cases. And you can also merge two or more graphs. And this merge process happens based on shared identifiers. And you can validate the graphs against the Biolink model, which is sort of a major requirement for us, at least in the translator project. And you can apply certain graph operations that you can. So how KGX does that is it loads the graph in memory, and then from there you can apply a lot of these cool operations that lets you do certain transformations on the fly, where you write or serialize that graph into one of the specific formats. So I just covered the model and the tools, but where is Biolink Model being used in the real world? We're using it in the translator, but outside of translator, or at least to give more concrete examples, there's something called as the SRI reference graph, which is a Biolink Model compliant version of the Monarch knowledge graph. This was built for the translator community, but it's also open to other, even outside the community. Monarch initiative, we have this graph contains ontologies like Go, HP, Mondo, Uberon, and also datasets like Biogrid, Reactome, ClinVar, Orphanet, and so on. And the graph itself is available at scigraph.ncats.io. It's still in rapid development, so we're still sort of constantly improving this graph. The next example of the use of Biolink Model is KG COVID-19, which is a framework and a knowledge graph. So the framework itself was built because we had a requirement of building a knowledge graph with data sets that were relevant for COVID-19 research. And this project was started earlier this year, sometime in March, April. And this is what we would call as the first instance of a knowledge graph hub. And the next is the Illuminating the Druggable Genome project, where we build a KG with datasets, specific datasets that are curated from papers and databases like Drug Central. And we're with the end goal being that we want to use this graph for machine learning, graph-based machine learning, with the goal of link prediction for predicting new drug targets. So to bring it all together, I mentioned the idea of a knowledge graph hub, which is the goal of the knowledge graph hub is to serve as a collective resource to simplify the process of generating knowledge graphs. And the idea being that we had to go through a lot of trouble and a lot of processes, and we had to look around to understand how to build KGs in a way that is easy to understand and easy to adopt. So the Knowledge Graph Hub is sort of a set of design principles and guidelines, and which allows you to build graphs that are easy to share and easy to work with other such graphs. And one of the design principles is using Biolink Model schema for the knowledge representation. And you can find more information about the Knowledge Graph Hub at the following links. And instances of the KG Hub can be found in the Knowledge Graph Hub organization on GitHub. So in terms of future directions, model itself is being constantly refined, and we have a lot of work ahead of us where we would like to add more things like better representation of variants and genotypes by looking at the genotype ontology, increase the expressivity of the model for things like evidence, provenance, context, and confidence by looking at the CPO ontology, and also better represent genomic locations using by looking at FALDO. And throughout the process, what we want to do is align to other ontologies, control vocabularies, and schemas, so that this becomes easy to go for at least users working with other schemas and ontologies to bring their data to the Biolink model. And in terms of contributing, the model itself is available on GitHub, and we have requests for improvements can be through GitHub tickets or issue tickets, and any contributions are welcome using through pull requests. And the documentation is available on biolink.github.io, and we also have a Gitter called the Biolink Model Community that is for anyone who wants to drop in and talk to us about their modeling needs, or if they just want to find out more about the Biolink model. With that, I'd like to thank the members of the Translator Consortium, Monarch Initiative, and the Berkeley Bioinformatics Open Source Projects. And also I'd like to thank all these people here, but more specifically, I'd like to give a shout out to Chris Mungall, Harold Solbrick, and Tessin, Matt Brush, Richard Bruskiewicz, Justin Rees, and Jim Balhoff, who are all on this call here with us today. And with that, I'd like to move forward with question and answers, or just discussions about what people have in mind and what their concerns are. Am I still here?
Yes, you are.
Okay,
Yes.
great.
Nice talk, Deepak. Thank you.
I thought I got disconnected for a second there.
Can you say something about if I had a knowledge graph and I wanted to use Biolink, do I have to be kind of fully conformant to the model and get everything absolutely right first time, or can I use kind of parts of it and maybe extend it?
No, I think that's a problem that we saw very early on, that it's not possible to be completely compliant. So what we recommend is you start simple and start focusing on small parts, and then refine that further iteratively. And that's how we've been doing with our other projects. And yeah, so it's not like you have to be compliant from day one. It's more of reconfiguring or adding more information to your knowledge, and then using Biolink model classes to sort of tag these different data points or your knowledge with the Biolink classes. Let's share screen. Okay, we're back.
Hi, Deepak. This is Sierra from the Alliance.
Okay.
And the Alliance is really interested in using the Biolink modeling language to model some of the new work we're doing to bring all the mods together. And I'm wondering how that, I guess, knowledge graph would fit in with Biolink, and how we would go about making sure that our knowledge graph fit in with the existing knowledge graph in Biolink.
Yeah, I think it also depends on the long-term goal on what is the sort of end goal you have in mind, right? And also your technologies that you use will matter a lot there. But yeah, I think one benefit I see there is, like, if you were to use Biolink model for representation, it just becomes a more intuitive style of how you would model different data sets. And we have enough tooling to support the adoption of the model, but it also lets you hook into other consortium-led efforts, and even outside of the consortium that is modeling data using Biolink model. But yeah, to give a more firm answer, I'd like to maybe talk more in detail about the needs of the Alliance and the kind of data types that you have, because at the end of the day, the model is very much at a higher level, and we don't prescribe just how all the nitty-gritty deals. The finer details about how the modeling should happen. It's just at a higher level, so that it's much easier to interoperate. So you're free to use your ontologies or other schemas, but the recommendation is to at least tag certain classes and associations or statements in a way that can be understood through the Biolink model dialect, which then helps us at least pull in parts of that we can understand and work with.
Yeah, so if we had like a gene, using your example, we would sort of link in our gene model to the Biolink model using particular syntax in the Biolink ML. And then we would be able to say in the NCATS or whatever, this gene object is equivalent to the Alliance version of a gene object. Is that sort of how it would work?
Yeah. So that's what we do with mappings. So in the model itself, we have this idea of exact mappings, which are where we can say that this term in the Biolink model can be used interchangeably with this term from that schema or ontology. But then we also have, there's a level of granularity. So we have exact mapping, we have narrow mapping, broad mappings that kind of captures the sort of essence of, to what level can these two terms be considered equal, or sometimes they may not be equal because there's some semantic differences there. So they may not be exact mappings. But to your example, yes, an exact mapping would say that the Biolink class would be a drop-in replacement for the Alliance gene.
That seems like really powerful, especially in the more complicated use cases of when you get to a little bit of a higher level object. I'm not saying it's easy to define what a gene is. We all know it's not. But like, for example, like a genotype or a strain, it seems like it would be nice to have that mapping in place, so you could sort of say that a genotype in Alliance is the same as a genotype, somewhere else, even in human, etc. So, and how are you using those mappings right now? What's the real big picture benefit of having those mappings?
The mapping, I think for us, at least for me personally, the experience that I've had is say I'm working with a separate a data set, even outside the translator consortium. I'm working with a data set that that talks about where there are already statements where we know that there are nodes of a particular type, and there are associations that describes how two nodes are linked together. But if they're using an ontology, say RO or something else, the mappings is what we use to bring or map these different concepts or terms to the Biolink model sort of vocabulary. And then from there, it's easy for us to then create outputs that are Biolink model compliant, right? So the curation happens manually, but then after that, it's easy the model, and then as part of your data transformation or ETL pipeline, you can always query the model and get back the relevant mappings that or get back the relevant Biolink mappings, which then you can use for your knowledge representation.
So would it be correct to say that at the moment the main use case for the mappings is more for humans to look at them and get a sense of how this model aligns with another model, but in the future it could potentially be used more computationally to maybe automate some parts of mappings or to automatically check for consistency between two schemas?
Right. Yeah. Long term. Yes. That is correct. Yeah. Right now, I think we're very hyper focused on improving the mapping and the readability of the model. But yeah, long term, we'd like to have this be more computable and also have do some consistency checks so that we know what mappings are valid at times and what are not. Because right now it is based on our intuition, but we'd like to automate that process. Yeah.
Deepak, Gustavo here, quick question. So I'm not really understanding the deep difference between association and predicate. So for example, looking at the list of predicates, I see is-missense variant of. And when I see such a predicate, it's kind of obvious that it has to relate to an association between a variant and a gene.
Right.
And I can think that, there are some other predicates that one could attribute to different types of associations, in which case it sounds like as if it would be like conflating different semantic connections. So in other words, I'm not really understanding why predicates and associations are not part of the same hierarchy, with multiple predicates being children of the same kind of association.
Yeah, I'm glad you bring that up, because there's a fine line between predicates and associations, because an association, it can be considered a sort of function of a subject predicate object triple. So we know something is a gene-to-gene association because we know the source or the subject node and the target or the object node is of type gene, and there is a link between the two. And the relationship there can be one of the many predicates that it can have, the predicate that's used as part of that association. So in a way, you are typing an edge, not just the predicate, the edge, the triple together to be of a particular type. And the default can be an association where you do not know what the more granular type of association that is. But more specifically, it would be easy to infer just by looking at the subject, predicate, and object what that association type is. So while, sure, the more information-rich part here is the predicates themselves, because that's what kind of explains the nature of the relationship. But to type that entire edge by itself, we sort of use association types from the model. Does that make sense, or did I gloss over?
No, I get what you're saying, but it still sounds to me like one could have designed this to be the same thing, to be one joint hierarchy of associations and predicates.
Actually, I think that one has to reflect on the fact that associations and predicates have two slightly different uses in the model, in the sense that an association is actually a full model of this, as Deepak is saying, the subject, predicate, object construct. And so you can actually hang a lot of different things off of that. You get slots and various slots that refer to other metadata or pseudo edges, as it were. It's kind of like a bit of a reification of the association. Whereas the predicate is very pure. It's just basically, in many cases, the predicate is closely aligned with association. But there's many of the predicates that can be used with multiple kinds of associations. So it's not like a strict one-on-one mapping of the predicates onto the associations. So I think keeping the two cleanly separated is important from a modeling perspective, and even from the semantics, it's not completely one-to-one predicates onto, or one, what would it be, many-to-one of predicates onto associations. It's more like a many-to-many relationship, right? The predicates are really just sitting in the middle as RDF, you know, in the spirit of the kind of RDF triplet subject. The predicate really has its own defined place, and so, and you can organize those predicates on its own independent hierarchy because of the semantics of the predicate alone, forgetting about the association. I don't know if that kind of clarifies things a bit.
I think one thing I want to highlight that is answering that. I think what I can understand is the idea of association is more relevant in the RDF world because you cannot type an edge using a predicate. That's not possible. And which is why we need something more in terms of the vocabulary. So if you were to consider, say, Neo4j and association types is just a property on that edge, which further describes that edge, that you may want that, you may not want that. But in RDF, you really need sort of a way of typing these statements so that it's easier to work with them and sort of bridging across between property graphs and RDF graph representation, because in RDF, statements are just statements. You cannot have properties on statements. And a way to get around that is what we call reification, where these statements are represented as nodes by themselves. And then, but then to allow for something like that, you need to be able to type that statement to be of a particular, you need to be able to say that statement is of a particular type. And that's why we have this sort of a separate set of hierarchy in the model to support for that.
Okay, thank you.
Yeah.
Hey, Deepak, this is Eugene from Tufts CTSI. I've got a question that's less about the model itself and more kind of about what's represented within it currently. So we have a specific use case in mind, possibly for the Biolink model. And we're wondering if you had concepts related to the PubChem database and the NCI genomic data commons. I just want to know if you're familiar with these resources and if you know if Biolink can model the data within them effectively. So if not, I can kind of try to explain, what we're kind of looking for.
So I personally have looked at the genomic data commons and their sort of idea of data dictionary and sort of data type a couple of years back, but at least it would be good for the benefit of everyone on this call if you can give an example of the type of data types you're looking into.
Sure. So, I mean, there's a lot of mutational data, for example. They have, I think they're called MAF files, or mutation annotation format, where they're basically just a summary of the mutations in a particular patient data type. They've also got, I believe, copy number variants and things like RNA-seq data as well, so expression data. And so part of this is just trying to create some engineered features around patient cancer profiles. And we were going to try to use Biolink in order to link up sort of the concepts that are represented within that data and apply it to treatments and compounds that might come from PubChem. So I just didn't know if you were familiar with those and if you knew how concepts in Biolink might be able to help us engineer those features. I
think there is some parts of what you said is still there in the model, but some not, which is why I think now is the perfect time for you to be involved in the discussion of how we represent variants and genotypes. And like I said, we are looking at genome as a way of modeling things, and it would be great to have your inputs there as well. Okay. Yeah, you can drop in for our help desk or just raise ticket, and we can definitely look into that as well.
Okay, thanks.
Deepak, this is Kimberly. Could you show your COVID-19 example again and
right here?
Yeah, and were there any specific, challenges or things that you had to overcome to put this together that would be worth maybe some of the primary data providers should know about?
Oh, yes. Chris, do you want to take this? Because I know you have strong opinions here.
Well, first of all, are we not talking more detail about KG COVID-19 later on, or?
We are, but I'm not sure if we would address those particular concerns that Kimberly just raised.
Yeah. So, but I'm sorry. What did you have in mind?
Oh no,
Deepak,
this
when
is
I had,
more about the identifier issues between UniProt versus the way these IDs are represented. There were some differences, and we had to manually hand curate them.
Right. Yeah, that was less of a modeling issue and more of a kind of like what identifiers should we be using for some of these entities, because there wasn't really a kind of like a clear set of kind of like single IDs that could be used for all of the different functional units of the SARS CoV-2 proteome. But that, I mean, I'd say that was less of a modeling issue, but maybe after we hear Justin's talk, we can talk a little bit more about some of the specific modeling challenges that we encountered there.
Okay, yeah. Yeah, so we do have maybe a 10-minute presentation from Justin Reese, who's here on the call, who will talk about his experience with Biolink Model and KG COVID-19, where we can probably dive deep into the more.
Okay, great.
Okay, then I think we can move on. So the next thing I have is sort of a demo where I will probably just step through a small sample data set and how sort of a thought process on how you would go about looking at the data and sort of using Biolink Model for representing that data set. And for everyone here on this call, I will share the link here in the chat. And this is sort of stepping through an example. And I'm sure there are more contrived examples that we can go into, but I'd like to start with something simple, that is modeling interaction data from StringDB, because almost everyone here is familiar with StringDB. This exercise, or this, at least this demo, is sort of split into 4 parts. You first look at how to model interactions, then how you map from protein identifiers to gene identifiers, and then represent the gene with its metadata, and then combining everything into a graph. The examples, what I'll be showing here in the slide, is available in this link here at github.com, Biolink Model Workshop. So StringDB is a database of known and predicted protein-protein interactions. And for the sake of this example, we'll just consider the protein links for a human. And the file, this file basically contains pairwise interactions between proteins with scores that kind of qualifies that interaction, at least the confidence of that interaction. But before I dive deep into the modeling aspects, I'd like to address a concern, which is genes versus proteins. We know they are separate, and genes encode proteins, and proteins physically interact and carry out the functions that they were designed for. But databases frequently use one or the other ID for representation. Some databases may annotate genes to describe certain protein or RNA activities. And in case of UniProt, there's this idea of a reference protein, especially for humans, where a single protein UniProt identifier can stand as a replacement for all the proteins encoded for that gene or that gene's isoform. So in the model itself, we try to bridge this sort of this kind of approach of this perspective by having a class called the Biolink gene or gene product, which is a sort of a parent of both gene and a protein. So now going to the StringDB example, this is sort of the original file. We have 3 columns, protein 1, protein 2, and combined score. And we know just by looking at it, since we are familiar with certain identifiers, we know that these are human ensemble proteins because of the 9606 that start with at the beginning of the identifier. And each interaction is qualified with a score. for modeling the data, we can make certain assumptions, and these assumptions are based on what you would like to see towards the end. What your graph should look like. So keeping that in mind, we can say each protein is a node in the graph. Each interaction is an edge in that graph, and each protein identifier can be prefixed with an ensemble prefix to have what we call a query. And the idea of having a query is because query or compact URI is a sort of it's a concept in the semantic web technology where the idea of linked data is that everything has a resolvable URI, where you can with a resolvable URI that describes that resource further. So the ensemble prefix is basically a short form for an IRI that it expands to, and then that IRI with the identifier together can take you to a location that will further describe that particular entity. So we want to be doing that when we're creating knowledge graphs, because we want these nodes themselves to be self-describing, and we want them to be pointing to locations where you can get more information about them. And the IRIs are usually defined by the original groups who are actually providing more information about these resources. So it's more upstream. And the 9606, it means it's part of the NCBI taxon 9606. And then if you click on that, it will take you to the bio portal, which will talk more about each of the that particular taxon and gives it hierarchy. So there's a lot more information. It's not just an identifier. That identifier itself has a meaning and why that representation matters. And so in Biolink model terms, we can say that each protein is of type Biolink protein. Each interaction is of type Biolink gene-to-gene association. Now this might be confusing because we're talking about proteins and we're doing gene-to-gene association. That's because in the model, what we mean by gene-to-gene association is basically a gene product to gene or gene product association. So we're conflating those terms together. And the predicate relationship that links the two proteins can be Biolink interacts with. And we can have an interaction score be represented as a property on this edge. And we can also have a provided by field that says who provided this particular statement, and that is string DB in our example. But it's not enough to just have, as I mentioned earlier, sometimes there is benefit in sort of translating protein identifiers to gene identifiers for the sake of, say, better merging or better representation. And so to achieve that, what we can do is fetch more information about the genes and get a mapping of gene to protein IDs, and then create something like a TSV that has information about where we say this protein maps to this gene, and the DBXref here is basically the NCBI gene ID. And I'm approaching this from a person who would go about writing a transformation pipeline and how the thought process would be in that regard. And again, keeping the graph in mind, we can say we have a node for a gene, another node for a protein, and ensemble prefix for ensemble IDs, and NCBI gene prefix for NCBI gene IDs. And the link between the gene and its product can be represented as an edge in that graph. Again, using Biolink model vocabulary, we can say that the link between a gene and its protein can be Biolink has gene product, and each gene can have an additional XREF, which represents cross references, can hold the alternate NCBI gene ID, which can then be used downstream if you ever want to do some remapping, or if you just want to have that there for reference. But the third part is it's not enough to just have genes by themselves. We want more information about the genes, their metadata like name, symbol, cross references, genomic locations. And there are many sources to get this information from. You can get it from Ensemble and NCBI. But what we use here is a service called the MyGeneInfo, which is an API that you can query using certain IDs to get back information about them. So what MyGeneInfo does is sort of aggregates information about certain biological entities. And we use the BioThings client, which is a Python library that can be used to query this particular resource. So even with this metadata, we can have all the properties that we get as properties of that gene represented in that graph. And especially genomic locations. We can represent genomic locations as a particular type of association called the Biolink genomic sequence localization. And the relationship that links a gene with its location is, gene with a chromosome or a genomic entity is Biolink has sequence location. So that's sort of how I would go about modeling all these additional information about the gene. And once we bring all these three data together, what we have is a combined graph that has representation about the gene-gene interaction, gene-to-protein information, the gene metadata, and the gene locations. And this is sort of what we would say is a TSV representation of the graph, where if you were to serialize a knowledge graph or serialize a Biolink knowledge graph, this is how it would look like. As a TSV, we have each column in the graph nodes TSV is a property of a node, and each row corresponds to a node in the graph. Similarly for edges. And excuse me. Okay, so what this allows, so this sort of representation makes it easy for exchanging graphs, and this sort of representation is very similar to what we would say, what we would see in Neo4j. So if we were to load this graph, this is how the graph would look like, where we have a central protein that links with, that interacts with other proteins. Each protein has their, central gene that links with other genes. Each gene has its own protein, and we have location information where the genomic coordinates themselves are represented as part of the edge. And I know I went by this very quickly, but each individual files at each step is available on the GitHub repo. And the final graph can be found here, where we have multiple forms of representations. We have the TSV and also N triples for those of you who are more inclined to use RDF, where the same graph is sort of serialized as N triples, which you can then set up a place graph instance or a Sparkle endpoint. But yeah, this is sort of how the thought process would work if you were to represent something, say from StringDB. And we sort of use the same approach for modeling data sets from many other sources. And for example, KG COVID-19, we had to work with many different sources. And Justin here will talk about more on how his experience has been with using Biolink Model. Justin, wait, does anyone have any questions? I know I went by there quickly. Okay, then Justin, do you want to then tell us about KG COVID-19 and how Biolink Model has helped in at least achieving the goals that we had for this project?
Sure. Can everybody hear me and see my screen?
Yep.
Okay. So I'll tell you briefly. Well, I think you said 10 minutes or so, Deepak.
Take your time. I just said 10 minutes for the sake of pacing, but yeah, take your time.
Okay. So I thought I'd tell you about KG COVID-19, which is our knowledge graph for COVID-19 response. I'll tell you up front what I'm going to tell you. You can read all about that here. I mean, so there's a GitHub repo. All of this is free and fair and open source, et cetera. We just published a paper about this if you want to know the gory details. So our purpose in putting this together, we're pretty good at making knowledge graphs. So when the pandemic hit, we thought that's something we could contribute, was a knowledge graph for COVID-19. And then specifically, our use case was to do drug repurposing after we'd make the knowledge graphs, to do graph machine learning and then find drugs that we could repurpose and suggest those as COVID-19 treatments. And sort of other use cases evolved too. So it's being used in the N3C project 2 and NBBL, as Deepak mentioned. But I thought what I'd talk about briefly today is our experience with sort of building a knowledge graph with the Biolink model baked in from the beginning, and you know, some things that buys you, and maybe talk a minute about this KG Hub concept that Deepak mentioned. Probably don't need to burn a lot of calories convincing you that knowledge graphs are a sensible way of approaching this, but the just briefly, so the idea is, even when before the pandemic hit, there was plenty of COVID-19 data around from SARS-CoV-1 and MERS, and there's plenty of other coronaviruses that are pathogenic and non-pathogenic, and then SARS-CoV-2 hit, and everybody started sequencing daylights out of everything, and there's all these bespoke, targeted data about SARS-CoV-2. And this is getting dated, but there were, I think last time I checked, which is a couple of months ago, there were 23,000 papers on this, and it's doubling every 20 days. And so it's probably more like 100,000 now. I mean, it's just a deluge of data. And so the lack of data is definitely not the problem. The challenge here was integration of data. And it tends, of course, it tends to be siloed and trapped in PDFs or behind APIs, and everybody has their own file format they represent the data in. And so integration really was, we think, the name of the game. So just to give you an example of that, like if you want to do something simple, what drugs are FDA approved? If you want to start on the code, on the drug repurposing thing, that's easy. It's probably just a Google search. What drugs or what proteins are targeted by those FDA approved drugs? That's maybe a little harder, but a couple files and a grep command and a couple of downloads, you probably have it. But then if you want to do something more useful, like what FDA approved drugs target a human protein that interacts with SARS-CoV-2, now you're getting closer to something that might be useful for drug repurposing. But then you're also talking about downloading several files and joins and merges and normalizing IDs and writing bespoke Python code probably, or some kind of code. And this is a case where knowledge graphs really shine. So that example I just gave is once you go to the trouble of making the knowledge graph, then it's really just some kind of graph query, like a Sparkle or a Cypher query. And this is an example of exactly that. And these are real things that came out of that preliminary analysis we did several months ago. And so we did actually go to the trouble of making the KG COVID-19 knowledge graph, and here's sort of a summary of what's in it. We have about 30,000 drugs and 30,000 chemical compounds, 50,000 or so publications, 40,000, the entirety of Go essentially for gene functions, all known human diseases, pretty much all human genes and human proteins, and all the SARS-CoV-2 genes and proteins, and a bunch of other coronavirus genes and proteins, and then human phenotypes. And this is all sort of done in an ontologically aware way. And so it's sort of the ontologies I just mentioned are sort of the scaffolding for the knowledge graph. But this is kind of closer to what I wanted to talk about, which is the sort of unexpected challenges with knowledge graph projects that probably a lot of people here have encountered. But it's once you go to the trouble of making the knowledge graph, it's harder than you would think to actually take stock of what is in the knowledge graph. And for example, like how many drugs do we have? That's, you can probably guess just with grep and some graph queries or things like that. But the Biolink model really makes that pretty easy. So it's harder than you would think also to interoperate and merge with other knowledge graphs. So, a lot of people spend a lot of time integrating data in other ways, and it's harder than you would think to, even if they're in knowledge graph form, to sort of merge or, compare and mix and match with other KGs. And sharing tooling between knowledge graph projects is also non-trivial. I'll give you a little spoiler. So but what the Biolink model gets you when you buy into it is access to these great tools like KGX dashboard, the dashboard that I'll show you in a second, the BNT and BML, and so all this great tooling that people like Deepak have been instrumental in making. You can leverage the efforts of all that great work. So yeah, that's kind of the rough pitch for the Biolink model in our use case. So I'll sort of talk for a minute about how we approached making the knowledge graph. So this, I think, as far as I know, Deepak will correct me, it was kind of the first knowledge graph where we sort of baked in Biolink compliance from the beginning, as opposed to sort of merging it into an existing knowledge graph project, which is certainly something you can do, and we've done it a few times now. this was meant to be a targeted knowledge graph. So our original use case, like I said, was drug repurposing, and so we tended to concentrate on drugs and drug databases and things like that, and obviously drug targets and PPIs and things that might be useful for drug repurposing. And we sort of designed this with the idea that we probably have to iterate on the ingestion. So we'll take this and that from this drug database, and then somebody says, okay, we probably need this other column from some file, so we can iterate on the ingest that way. We're only taking in authoritative data, so if we're ingesting a drug database, we're not going to take their word about PPIs. We'll get that from string or intact or someplace like that. The idea, well, I'll show you this in a second, is sort of download and transform first, and then make a graph, sort of a subgraph of every ingest that we do, and then we can remix it with the tools that I'll describe in a minute. This is kind of another strange thing when you make a KG project that has an ETL component, which that's largely what this tooling is about. Is downloading is a very frequent point of failure, and so we have some tooling that kind of addresses that, and sort of one of the things we do is sort of cache the downloads. And so if the download fails for some reason because somebody moved a file or renamed it or something, which happens all the time, then we can go back to the last time we downloaded it and use that sort of cache of data. And ideally, that's all the network dependency is early on. So in theory, once you have the downloaded files, you could unplug the Ethernet cable, and this would also work. ID normalization obviously is hugely important for a KG project like this, and I won't go into great detail, and I probably don't have to convince anybody. If not, then drug bank and then PubChem. So also the idea is to sort of deliberately do ID normalization during the ingest. So sort of put that onus on the developer who's writing the ingest to very deliberately do this ID normalization. There's a few tricks we can do after the fact once we have the subgraphs, thanks to Deepak's work on KGX, but mostly this is something we sort of want the developer to do during ingest. So here's sort of an overview of how this works. And this essentially, like I said, is more or less just a tricked out ETL pipeline. So these are the data that we want to download and ingest. Like I said, this is hopefully the only network dependent part of this tooling. Download the files, cache them somewhere. The transform step is next, and like I said, this is sort of so we're baking in the Biolink model compliance here, and this is where that happens. So every source that we want to ingest has bespoke Python code that does that and produces a subgraph that's kind of stored separately in that format that Deepak just described, the KGX TSV format. And these are sort of the component data as separate graphs. And then the merge step, which basically leverages KGX to do most of the work, can combine the component graphs in a YAML-driven way, and does click merging kind of as it's doing this, but mostly the ID normalization would take place during this step, but there's a bit of that takes place in the merge step too. Another point here is that thanks to KGX, we can mix and match these subgraphs in a very fine-grained way. And so if this use case, or, for our machine learning use case, we want this one, and this one, that's easy to do with KGX. If somebody else comes along, they want everything we have just in a separate merge file, you can merge YAML file, you can describe that, and that's easy to do. Like I said, when you do these KG projects, it's harder than you would think to do something trivial like ask, how many drugs do we have, how many phenotypes do we have, or things like that. And the Biolink model makes that dead easy. You don't actually need fancy displays to do that, but Kent and Deepak has spent some time making this really slick dashboard, which is something you get if you sort of align with this KG Hub way of doing things, which I'll describe maybe a little bit more in a minute. Here, I'll show you. This is the live site. So if you do things sort of in a KG Hub way, which the canonical examples of that, then you get this tooling for free. And I think Deepak, but this kind of should work out of the box if you use our tooling, take the knowledge graph, and so it's dead easy to figure out. We've got a lot more publications than we used to. 129,000 publication nodes. 11 million or so interacts with those, and so on and so forth. You can drill down in a pretty sophisticated way to see what exactly is in the knowledge graph. It's a nice Sankey plot which shows you, what Biolink categories are coming from each source, and so on and so forth. Let's see. So it's not, the Biolink model is not just useful for taking stock of what you have. You can use it in downstream. It's useful for a lot of downstream things too. So for example, like I said, the next step after we made our knowledge graph was to do graph machine learning to find drug repurposing candidates. The first step of that was kind of to take stock of to embed the graph, which I won't go into, but then sort of see what the graph structure looks like in a t-SNE plot. So we make the embeddings and then display what the nodes look like in a t-SNE plot, and the edge embeddings look like in a t-SNE plot. So conceptually, this is basically just taking the embeddings, which are like a 100-dimensional vector that represents each node. And so things that are close together in a t-SNE plot are kind of more associated. They're in the same network neighborhood. But it's useful, for example, to plug in the Biolink categories when you're doing the node embeddings, just to see, where did the publications end up. That's this blue smear here, and then you can see the ontology classes that are mentioned in those publications are smeared along with it. And so this is sort of the associations that we're leveraging to do the graph machine learning later on. I'll just say briefly, this is kind of unrelated, but we made this knowledge graph sort of for our drug repurposing use case, but it turned out to be useful for a bunch of other things. We sort of passed this on to the N3C, the National COVID Cohort Collaborative, as a way of organizing they have more clinical data than we have, but they found what the KG to be useful as sort of a scaffolding to put all the public, so to speak, public COVID-19 data and the ontologies in there, and then put the clinical data on top of that, sort of as a scaffolding. And I'm sure Melissa was on the call. She's done a lot of great work to make this sort of democratize this data and make it easier for everybody who wants to take advantage of this resource to actually get access to the N3C data, which is several 100,000 COVID patients. Well, she'll correct me on the numbers, but a lot of really great data that now hundreds or thousands of researchers can now use. So Deepak also mentioned the NVBL, which is the National Lab effort for COVID-19 response. We've used this in a couple of different ways for that project. Was sort of prioritizing drug targets, and so we've used this for hypothesis-based querying to identify possible drug targets. There's also this cross-cutting aim, which is more about integrating data from different efforts. So and KG COVID-19 was used to sort of drive, well, as one of the ways of displaying integrated COVID-19 data that researchers can take advantage of. So how am I doing on time? I wanted to really briefly mention that this is sort of a related concept, which is the KG Hub concept. So I mentioned before, this is kind of what we have now, which is kind of a tricked out ETL pipeline. And what you end up with after the transform step is these local, these subgraphs that represent any given ingest. Conceptually, we have all the pieces in place to mix and match data, not just from local graphs, but from remote graphs. And so there's a couple of advantages here. Is it's if other projects, and we have 3 or 4 now, align with our way of doing things, and it's dead easy to know what data exists remotely. So you know, if you go looking for gene to disease data, it's kind of not trivial to do. I mean, you have to do that in a kind of deliberate way, but the more KG projects we have aligned to the KG Hub, the easier it is to sort of just to easily find, for example, show me all the gene to disease data, and so on and so forth. And it also, all the tooling we have it makes it dead easy to sort of merge and mix and match. And so I'll give you graphically what my idea of this KG Hub concept is. Well, our idea, I should say. So these are sort of projects I would say that we have that are loosely aligned, and you can use a lot of this tooling and sort of way. There's the IDG project, Monarch project, KG COVID-19, and we'd encourage other people, if they're interested in aligning to this way of doing things, we definitely want to work with you on that. And so the wins here are reducing the duplication of effort on ingests, because these are not trivial to write. And they take a lot of developer time and a lot of care and feeding to keep them working. Shared tooling, so KGX is a tool that I think is really great and is super useful for these kind of projects. The dashboard I just showed you, a bunch of other things I'm probably forgetting. And then another win here, and this I wouldn't say is sort of a work in progress, is this kind of allows a cafeteria style remixing of KG content. So, you know, all this effort that we put into making all these knowledge graphs can be reused. You know, if another pandemic hits, or there's some other use case to do with cancer or something, a lot of the same data is going to be the same. Like you probably are going to, string would be useful. You're probably going to need PPIs. You're probably going to need drug data, you know, gene to disease or variant disease if you're looking at cancer, and so on and so forth. So we think this is a useful architecture for allowing a lot of these wins to happen. So I think I'll stop talking there, but if anybody has any questions, glad to answer them.
All right, thank you, Justin. Yeah, does anyone have any questions about KG COVID-19, or just in general? I think we can open the floor to everyone and maybe talk about if they have any questions on the previous presentations, or just in general, their concerns about other models or other schemas, and maybe we can look into that, or their use cases on what they would like to get at with Biolink Model, or see ways how we can align other schemas with the Biolink Model. So, yeah. Okay, I'm guessing there are not many questions then.
Well, I was going to mention I'm on the phone with my supervisor as well. Justin, are you? I'm just a little confused here. Are you with the translator group, Justin? I'm just.
No, I mean, I sort of work peripherally with a lot of the people on translator, but I don't think I'm officially on that project.
I don't want to take up too much time here, but my group is wanting to do something similar to what you've done with the COVID-19 data. We have other applications and trying to align it with Biolink. I was just wondering if we could maybe meet up later, schedule another time to discuss. We kind of want to see if you could share some of your wisdom with how that was done. Okay. Is there a way I can reach out to you? Since you're not in translator, I don't have any contact information for you.
Let me.
I can post my email in the chat.
Actually, believe it or not, you can find Justin online in the Slack for translators. So just
Oh,
connect.
really?
Yeah, I just found him there.
Oh.
Oh yeah, I
All
think
right.
I did an onboard. I haven't done a lot of work with the translator people.
But Justin has been part of previous translator relay sessions which was more COVID specific. So Justin has been part of that discussion as well. So yeah, definitely we can connect through Slack or and set up a meeting as per our needs.
All right, thank you. I'll reach out to you on Slack then.
Okay, great.
Thanks.
I have a question about the KG hub concept. Are you thinking of all of these KGs being in KGX file format for sharing, or are you just thinking of more sort of a loose kind of integration through, say, a web services application? Okay, so what do you have in mind in terms of this KG hub?
Well, I think one of the organizing principles that Chris kind of espouses, and Marcin, who's on the call, I think has done some work on this too, is we're not kind of about enforcing rules. I mean, there's really just what we had in mind, at least at the beginning, was sort of a loose confederation of KGs. It does, I would have to say, it probably would make it a lot easier if people did use KGX TSVs, or at least make that format available for the mixing and matching, because that way the KGX can kind of have an easier time of working with it. Yeah, I don't think there's any hard and fast rules here, and we definitely want to work with people who want to sort of, I mean, I think we can sort of take baby steps towards a complete alignment of KG projects. It's not an either or thing.
Okay, so I get the sense it's a multi-layered thing in terms of its activity. I was thinking more in a practical term, what's the end product here? Is it going to be like an archive, like a physical location where people would go and pull out knowledge graphs, or just a site that says, oh, if you want this knowledge graph, go here, knowledge graph, go there. So it's more of an informational site, or something in between. I mean, I can imagine many things that could feed into that. For instance, in Translator right now, we are discussing very actively how we're going to integrate and index KGs, mainly in KGX format, but sort of indexing in terms of metadata. How do you find what's in there? And we'll build some active infrastructure to support that, like a registry. So I just wondered if anything, any of that feeds into. So at the moment, I guess Knowledge Graph Hub is basically a repo, I guess. That's what you're saying, I guess.
It's a GitHub organization and
okay.
a collection of repos. Yeah, I think that would be what I would advocate, the way of doing this. I mean, ideally, this isn't practical, I guess, for some existing projects, but new projects could be repos in this in this GitHub organization, which would make, as you said, mix like sharing of files and just tighter integration. We have a lot of tooling that integrates with GitHub, and, Jenkins workflows that you could reuse. So I think ideally, yeah, it would be sort of integrated in this way, in this GitHub organization, but definitely that's not written in stone.
I mean, one model is to sort of take inspiration from OBO, Open Bio Ontologies Foundry, which on a kind of minimal level is really just a kind of a registry of files, ontology files in a standard format with some kind of guidelines as to, how to be a good OBO member, with varying degrees of conformance, between all the different members. But even just having that minimal layer allows people to build common tools. And we now have sort of common browsers like OLS and Bioportal and Ontobee that allow people to kind of search across all of these. And the overall goal with OBO is to kind of link these ontologies together. And there's some that do link well together, others link less well. And so the idea is to kind of really, one idea would be to take that concept and apply it for knowledge graphs as well, and really provide that kind of like that minimal layer of standards, and just allow the community to kind of build tools around that.
Right, and also to extend a bit more to that is that each sort of instance of the Knowledge Graph Hub can have any kind of their graph in any form, but we recommend that they also provide KGX in addition to KGX file formats or TSV in addition to what they have, just so that we can have that sort of interoperability with other graphs. And Richard, you mentioned something about a registry in Translator, which is different because that is just a registry for indexing different graphs and their locations. The Knowledge Graph Hub itself is a place for not just the graphs, but also how they are built, the sort of ETL process, the data transformation that happens. That code also lives as part of that repo, which is from that perspective, it's more of a heavy lift versus just a simple registry. That's also a sort of a clear distinction between the two.
For sure. I was just seeking clarification for the
Sure.
total vision here. Yeah, that's good.
And we do have sort of guiding principles here. I put the link here in the chat, sort of how what a Knowledge Graph Hub is, but this is still we're still refining that and improving that. That's sort of we have set of guidelines that you can use and set recommendations on how you should be modeling, the tools you should be using, how you should be versioning, and so on.
So I just wanted to say first, thank you for this presentation. I think the interest for the integration and querying of heterogeneous data sources is clear. I wanted to know, however, if you could touch base a little bit on prediction, and especially I would say in maybe custom prediction. What I mean by that is because we have a graph structure and because you have a common vocabulary, it seems that we could design some kind of very specific prediction, let's say to detect but something then that could actually be reusable as long as we conform to the Biolink model. So could you touch base a little bit on that?
Well, I can't help here, but plug another project Deepak and I have been working on called NEAT, which is an effort to kind of, I think it's what you're getting at. And so this touches on some other projects like Embiggen, which is our graph machine learning library. But essentially what it is Deepak and I's effort to sort of make it dead easy to do reproducible, easy YAML-driven machine learning on graphs. And my interest specifically for drug repurposing. So there's a lot of kind of mundane things that become kind of a pain when you do iterative machine learning on graphs, which is like, oh, how did I do that? What hyperparameters did I use? Where's that one file? And so this way, Deepak, would you mind putting a link to NEAT in the chat?
Oh, Chris just did. There's
oh, great. Yeah, cool. So the idea is you could do reproducible machine learning. You just have the YAML file that describes exactly how you did it, what the file's name, where it is, all the hyperparameters, the exact graphs and holdouts you used and everything like that. And so is that what you mean by prediction, Laurent Philippe? Like specifically drug repurposing prediction candidate?
Right. So it's like really when I have a very specific question, like what could be the drug target, for instance, for a brain disease, or maybe for something else, like, I don't know, if a protein like is part of a major protein complex, and can you maybe predict like which would be the missing elements of that complex? Like some very specific questions that I would say would require like custom prediction, but because we are using the common vocabulary, maybe that's something that can actually be achievable. Absolutely. Across resources.
Yeah, I would love it if NEAT evolved into that, and I think it's pretty much working. There's a lot of work left to be done to make that dead easy, but I think we have the tools to do exactly that. And yeah, and definitely if you have a use case that's similar to that, or we could build that into NEAT.
Yeah, I think that particular use case would really fall under link prediction, which I think is already covered within NEAT.
Yep, that's right.
How well it would work would depend a lot on the quality and size of the underlying data, but in principle it could work.
Okay, any more questions or comments or concerns? It looks like, I
Guess
think
we're
we're,
gonna get a lot of them in the future.
I hope so. Yes, I would appreciate, if not now, then at some point you can find us on GitHub or the Gitter community Slack where you can ask your questions and we can have the discussion asynchronously, and that would be great. Well, then I think we can probably end the workshop, and I'll hand over the controls to Sue for any closing statements.
No, just thank you all. Thank everybody for attending. It's been a great workshop. And hopefully you will go play with the Biolink model and then bombard Deepak and everybody else with questions. I think I have all the links that were mentioned in the Slack channel so that you can go back and retrieve them later.
Thank you.
All right, everybody, have a lovely weekend.
All right.
Thank you. Thank you, Deepak.
Thanks,
Thank you, Justin.
Sue. Bye.
Bye.
Thank you.
Thanks, bye.